Welcome back to the deep dive. Okay, so imagine your DNA, right? It's like the ultimate biological hardware. The fixed blueprint for, well, everything that makes you. But
Mhm.
here's where it gets really wild. That hardware, it comes with this dynamic, incredibly sophisticated software layer.
Right.
And this software, it doesn't change the blueprint itself, but it dictates how that blueprint is read, what parts are active, what parts are silenced.
Oh,
We're
okay.
we're talking about epigenetics, and honestly, it's way more active and responsive than you might think.
It really is.
So today we're diving deep into this amazing world. We're going to zoom in specifically on these cellular switches known as novel histone modifications and DNA methylation.
Yeah, the real control knobs.
We've dug through a ton of research here to pull out the most important stuff, give you some surprising facts, those really satisfying aha moments,
Yeah.
all about how these tiny cellular changes have these huge impacts on our health, our vulnerability to disease. It's kind of a shortcut to understanding your body's hidden OS.
And that's really the key thing, isn't it? We're looking at a layer of control that decides how our genes get used, not changing the genes themselves.
Right.
It's incredibly flexible, this system. It's constantly responding to, well, life's experiences.
Hmm.
And that responsiveness, that's exactly why figuring it out is so vital for our health.
Okay, let's unpack this then. This journey, understanding epigenetics, it didn't just happen overnight.
No, definitely not.
It's a story of scientists kind of building on each other's work over decades. It actually started, surprisingly, quite a long time ago, laying the groundwork, the concepts. I mean, think about it. Way back in 1942, Conrad Waddington first coined the term epigenetics. He was trying to explain this fundamental puzzle, how one genetic blueprint, the genotype, gives rise to all the different traits you see, the phenotype.
Huge question.
Then later, in 1957, he came up with this idea of the epigenetic landscape, sort of suggesting that cells differentiate, how a stem cell becomes a liver cell or a brain cell, not just by changes in genes, but by navigating different paths on this, like cellular map.
It was a really profound conceptual jump. And for decades, you had scientists like Nanny, Riggs, Holliday refining this idea, stressing how gene function could change and even be inherited
Yeah.
without changing the actual DNA sequence.
Right, without altering the code itself.
Yeah, and then Bird defined it more around adaptive changes to the chromosome structures.
Hmm.
But the big question hung there. How did we get from these sort of abstract ideas to the actual molecular switches we can see today?
That's the exciting part.
It took a whole series of critical discoveries, didn't it? Moving
Yeah.
it from theory to something tangible, observable.
Exactly. It was like slowly, piece by piece, pulling back a curtain. Think about the breakthroughs. 1964, the first hints that these tiny tags on histone proteins, the spools DNA wraps around, that they could regulate how genes are read. Then, just a bit later, 75, the discovery of a key DNA modification, 5-methylcytosine, like a master switch directly on the DNA.
That was massive.
Absolutely huge. Then in the 90s, we started identifying the specific enzymes, like the HATs and HDACs, that actively write and erase these histone tags.
The writers and erasers.
Yeah, these weren't just random facts. They were completely changing the picture, revealing this super dynamic layer controlling our whole genetic instruction manual.
And the pace just picked up in the 21st century.
Totally accelerated. Early 2000s, discovery of enzymes, adding and removing other kinds of histone tags, and for real-world impact, 2006 was big. FDA approved the first epigenetic drugs for cancer.
Showing it was treatable.
Exactly. These weren't just theoretical switches. We could actually flip them therapeutically. Then, 2015, the NIH Roadmap Epigenomics Mapping Consortium released this incredible resource 111 reference, Human Epigenomes, just freely available.
A massive data set really pushed the field forward.
It's amazing how fast our understanding grew. So let's zoom in now on one of the biggest players in this toolkit, DNA methylation.
Okay.
This is where it gets really precise, because it's not just about on or off, but how and where that happens with incredible specificity.
Right. Fundamentally, DNA methylation is just this tiny chemical tag, a methyl group, added right onto the DNA molecule, usually a specific spot's called cytosine bases.
Like a little chemical sticky note.
Exactly, like a cellular sticky note. And its main job, regulating gene expression. It can act like a do not disturb sign, basically recruiting proteins that silence a gene.
Shutting it down.
Or it can physically block the machinery that needs to read the gene. And this precise mechanism leads to these stable, tissue-specific patterns of gene expression. That's why heart cell stays a heart cell,
Makes sense.
And what's really cool is that specific proteins act as readers for this methylation. Proteins like MESI-P2, they're designed to spot and bind to methylated DNA, helping not just interpret the signal, but actually maintain it, keep those instructions stable.
You said where it happens is key.
Absolutely. Location is everything. If methylation happens in these specific zones called CPG islands, often near the start switches, the promoters of genes, it usually represses gene expression.
Turns the gene off.
Pretty much explicitly turns it off. But here's a twist. Lots of methylation within the main body of a gene is often linked with active gene expression.
Oh, interesting. So context really matters.
Completely. And maybe even more critically, DNA methylation is vital for keeping quiet these jumpy bits of our DNA.
The retrotransposons.
Yeah, exactly. Things like endogenous retroviruses, ERVs, they make up a huge chunk of our genome. Methylation normally suppresses them, keeps them from moving around or getting expressed.
Because if they get reactivated,
it can cause problems, genetic instability, or even trigger an unwanted immune response, like a cellular false alarm going off.
Wow. Okay, so connecting this back, what does this all mean for, for us, for our health?
Well, it tells us that our lifestyle, our environment, they aren't just out there. They're actively writing and rewriting this cellular software.
Yep.
Yeah, the sources point to things like air pollution, certain chemicals and plastics, heavy metals like cadmium or mercury, tobacco smoke. Even things like chronic stress or a high-salt diet. They can all leave measurable marks on these methylation patterns.
That's kind of mind-blowing.
It is. And to give you a really concrete example, some studies have looked at individuals who experienced severe childhood abuse and later struggled with suicidal thoughts.
Okay.
They found higher DNA methylation in a specific gene in the brain, one involved in the stress response system. This specific epigenetic mark might influence how effectively that person regulates stress later in life.
So early experiences literally get written into our cellular software.
It certainly looks that way. It raises this really important question, How much of our biology, our response to the world, is shaped by these experiences encoded epigenetically?
And it's not just stress, right? What about diseases, like, say, lupus?
Right. Systemic lupus erythematosus, SLE. It's a classic example in epigenetics research. Early on, scientists noticed that T cells, key immune cells from patients with active SLE, had significantly less DNA methylation overall.
A global reduction.
Exactly. And here's the crucial part. Drugs that inhibit DNA methylation, like 5-azacitidine, could actually make normal T cells become autoreactive in lab experiments.
Like they started attacking the body's own tissues.
Precisely. And they could even induce a lupus-like disease in mice just by causing this hypomethylation.
Yeah, wow, that's a direct link.
It strongly suggests a causal link. It seems like in SLE, certain immune cells might be, you could say, epigenetically poised, ready to overreact to inflammatory signals because key genes aren't properly methylated, aren't kept in check.
So these tiny methylation changes have these massive consequences for the immune system.
Huge consequences.
Okay, so beyond DNA methylation, there's this other really intricate layer of control, the histone code, right?
That's right. These are modifications happening not on the DNA itself, but on the histone proteins that DNA wraps around. Post-translational modifications, or HPTMs.
Like punctuation marks in the genetic script.
That's a great analogy. They add another layer of information, dictating how genes are read. If we think bigger picture, these tags on histones essentially control how accessible the DNA is.
How tightly packed it is.
Exactly. How compacted the chromatin, the DNA protein complex is. That ultimately affects which genes can physically be reached by the transcription machinery and turned on or off.
And when this goes wrong?
When it goes awry, you often see it linked to diseases, especially cancers. It's not just about which genes you have, but how they're packaged and regulated. And these histone modifications are the master controllers of that packaging.
So let's dig into some of the really interesting, maybe newer HPTMs, the ones that show this deep, almost unexpected link between, like, our metabolism and our epigenetics.
Yeah, that's a fascinating area.
First up, histone alactylation. This is wild because lactate, we mostly think of it as muscle burn, right? Metabolic waste.
The byproduct of intense exercise, yeah.
But it turns out it's way more than that. It's actually a potent signaling molecule, especially in tumor cells. That whole Warburg effect thing where cancer cells use fermentation.
Like they metabolize glucose differently.
And get this, the lactate buildup directly triggers this histone lactylation, and that profoundly changes gene transcription, especially in cancer and inflammation. It basically gives cancer cells an advantage, helps them grow, fuels inflammation around them.
So the waste product becomes a signal.
Exactly. And it's not just cancer. High levels of this histone lactylation are seen in severe sepsis, reflecting illness severity. And after a heart attack, it actually seems to boost genes involved in repairing the heart muscle.
Incredible. Who knew lactate was so multifaceted?
Right. Then there's histone citrullination. This one's done by specific enzymes called PAs. It's a pretty significant modification, actually.
Hmm.
And it does a lot. It's critical for embryos to develop properly. It's essential for tissue regeneration they've seen in zebrafish. It's key for wound healing. And it's even linked to aging, with certain citrullinated histones piling up in older cells.
Okay, so quite widespread functions.
But here's the real aha moment, especially for immunity.
Okay.
Histone citrullination is pivotal in forming something called neutrophil extracellular traps, or NETs.
Ah, yes, nets, the immune system's sticky traps for pathogens.
Exactly, they're crucial. But when net formation goes haywire, it's implicated in really bad stuff like sepsis. They've even seen increased net debris in severe COVID-19 patients.
So dysregulation is dangerous.
Definitely. And this citrullination process itself is strongly linked to cancer progression, too. It's being looked at as both a marker and maybe even a therapeutic target.
Interesting.
And one more quick one, histone 2-hydroxyisobutyrylation, or HIB.
HIB?
It's different from other tags. It adds a hydroxyl group, physically changes the histone shape, affects interactions. The thinking is it comes directly from metabolism, from a molecule called HIB-CoA.
Another direct metabolism epigenetics link.
Yeah. It lets cells basically tweak their gene expression based on the levels of this metabolic molecule. It just shows how deeply intertwined these systems are.
It really paints a picture of the cell constantly sensing its metabolic state and adjusting its gene expression accordingly.
And we should remember, these histone mods also do fundamental stuff like DNA repair, super critical, especially for the kind of DNA damage seen in cancer.
Right, organizing the repair machinery.
Yeah, and they're involved in meiosis too, shuffling genes during reproduction, which is essential for genetic diversity.
So they're fundamental cellular regulators.
Absolutely.
Right.
Okay, so spinning this forward, what does all this mean for actually treating diseases? This is the really exciting part, right? Because unlike our DNA code, these epigenetic marks, they're often reversible.
That's the key therapeutic promise. It makes them incredibly attractive targets for new drugs.
So what are we seeing there?
Well, in cancer treatment, the development of epigenetic drugs is a major story. You have the DNA methyltransferase inhibitors, DNMTIs like azacitidine and decitabine.
Used for blood cancers, right.
Exactly. Widely used for myelodysplastic syndromes and certain leukemias. They work by basically removing the enzymes that add those methyl tags.
So they lift the silencing.
Precisely. They allow previously silenced tumor suppressor genes, the body's natural brakes on cancer, to be turned back on. It's like reactivating the cell's own defenses.
And what about combining them with other drugs?
That's a really important strategy, sometimes called epigenetic priming. Using DNMT inhibitors can make cancer cells more sensitive to other treatments, like chemotherapy or targeted agents,
How does that work?
by demethylating or reactivating key genes that might make the cell vulnerable to the other drug. And in immuno-oncology, there's this fascinating angle. DNMT inhibitors can actually enhance the effect of immunotherapy. They seem to do this partly by reactivating some of those jumpy ERV sequences we talked about.
The endogenous retroviruses.
Yeah. When those get turned back on, they can trigger an innate immune response within the tumor, basically flagging it for the immune system, boosting anti-tumor T cells, making the cancer more visible.
Wow, that's clever.
Yeah.
Using the cell's old viral DNA against the cancer.
It's a really neat mechanism. And beyond the DNMPIs, there are also the histone deacetylase inhibitors, or HDACs.
Targeting the histone tags now.
Right. Drugs like fornostat and romidepsin are approved for certain lymphomas,
Yeah.
with many others being tested. They target different epigenetic pathways. And we're also seeing early work on inhibitors for those citrullination enzymes, the PAD inhibitors, potentially for stopping cancer spread.
So it's a growing arsenal. And it's not just cancer, is it? This has broader implications.
Oh, absolutely. Epigenetics offers insights and potential treatments for a whole range of conditions.
Like metabolic diseases. You mentioned the links there.
Definitely. Things like type 2 diabetes, obesity, non-alcoholic fatty liver disease, or NAFLD, they're increasingly linked to specific histone modification patterns.
And can we target those?
Potentially, yes. For example, the common diabetes drug metformin has been shown to reverse some specific histone marks. And drugs like decitabine, initially for cancer, are being explored because they seem to influence immune cells in metabolic contexts, potentially restraining insulin resistance or reducing fat buildup in the liver in NAFLD models.
So the reach is really broad. What about something like depression?
Another area with strong epigenetic links. Chronic stress, a major factor in major depressive disorder, MDD, can literally precipitate changes in histone configurations in the brain.
Really?
Yes, and interestingly, MTD has been correlated with reduced levels of certain histone tags, like histone crotinylation.
And are there potential treatments emerging from that?
There's some really intriguing, maybe surprising research. For example, bot-hydroxybutyrate.
That's a ketone body, right? From fasting or keto diets.
Exactly. It's been shown in some studies to have antidepressant-like effects. It seems to work, in part, by increasing levels of certain histone modifications and boosting brain-derived neurotrophic factor, BDNF, which is vital for brain health and resilience.
So our metabolism, through things like ketone bodies, can directly influence our brain's epigenetic state and potentially our mood.
It really underscores how interconnected everything is: diet, metabolism, epigenetics, brain function.
Okay, this is all incredible, but how do scientists actually see all this? How do they read this complex epigenetic software? The technology must be pretty amazing, too.
Oh, absolutely is. The technological leaps have been just as remarkable as the biological discoveries. We're not just looking at single genes anymore.
Genome-wide, right.
Exactly. Techniques like ChIP-seq let us map where specific histone modifications are across the entire genome. Others like DNA-seq and ATAC-seq show us which parts of the chromatin are open or accessible for reading.
So you can see the active regions.
Precisely. And newer methods like CT-INTAG and UCT-NERYUN are even more sensitive and efficient for mapping where proteins bind. Then there's mass spectrometry.
What does that add?
While sequencing tells you where marks are, mass spec can tell you how much of different modifications there are, and specialized techniques like MALDI imaging mass spec can even show you the spatial distribution of these modifications directly within a tissue slice.
So you could see, like, how a histone tag is spread across a tumor versus normal tissue.
Exactly. For instance, seeing increased H4K16 acetylation, specifically in liver cancer tissue, it gives you this incredible molecular map of the disease state.
And then there are the epigenetic clocks. This is genuinely mind-bending stuff.
It really is quite something. Researchers found that they can look at patterns of DNA methylation at specific sites across the genome and estimate a person's biological age.
Which can be different from your chronological age, right?
Significantly different, yes. These clocks, like the Horvath clock or the Hannum clock, act as biomarkers of the aging process itself.
What do they tell us?
They reveal fascinating things. For example, studies show that exposure to significant adversity early in life can predict accelerated epigenetic aging in children.
Meaning their cells look older than they should.
Effectively, yes.
Yeah.
And this accelerated epigenetic aging might increase their risk for various diseases later on. It gives us a potential tool for prevention science, identifying kids at higher risk. Maybe even measuring if interventions are helping to slow down that clock.
That's powerful.
It is. And all these advances really rely on collaboration. Big projects like the NIH Roadmap Epigenomics Consortium, putting all that data out there publicly, have been crucial for moving the whole field forward.
So as we kind of wrap up this deep dive, it feels abundantly clear that epigenetics isn't just some, obscure scientific niche.
Not at all.
It's fundamentally the dynamic operating system shaping our biology. It influences everything: immunity, metabolism, our risk for cancer, diabetes, even depression. It's this constant, really intricate conversation happening between our genes and our world.
And that's the really profound take-home message, I think. The sheer plasticity, the responsiveness of these epigenetic marks.
They're not set in stone.
Not at all. They're constantly being written, erased, rewritten by our environment, our diet, our stress levels, our experiences. And this doesn't just give us deep insights into how diseases happen.
And also how to fix them.
It offers incredible potential for new therapies that are potentially reversible. Unlike changes to the DNA sequence itself, the epigenome offers this amazing window for intervention, maybe even prevention.
So thinking about this framework, this epigenetic software, it really empowers you, doesn't it? Knowing that your environment, your food choices, how you manage stress, they're not just external things happening to you. They're actively involved in editing your body's instruction manual. It's like right now. It's a powerful thought.
It really is. And it leaves us with a pretty big question to ponder, doesn't it? If our experiences, our exposures can literally reshape how our genes are expressed without ever changing the DNA letters, what responsibilities do we have, not just for managing our own epigenetic landscape, but potentially what impact might this have down the line for the health of future generations?